remaining parts, called the exons, end up joined together and give you the actual coding region of that RNA. That's going to be read again as the polypeptide. So if you left these introns in, okay, you have a bunch of extra amino acids possibly, but the introns in fact could contain stop codons. They could cause a frame shift relative to what's read from just the exons. So it's important that they be spliced out. Okay. And in many cases, this RNA splicing is carried out by something called the spliceosome, which again is a variety of proteins and RNAs actually together. And they recognize what are called splice junctions. So they recognize the domains at the boundary between exons and introns, and then they carry out a very particular specific reaction so that they can splice out that piece of RNA, okay, and link together the two exons, okay, across each intron. Okay. And we know a little bit about the sequences that are involved in this. Don't worry about trying to memorize this. But we know the nucleotides that are normally seen at a splice junction. But it's not so easy to predict where these are. Okay. Because these sequences are not that specific. And so usually the way we know where they are is because we actually have sequenced both the DNA and the RNA. We know what part was transcribed, and we can see which parts must have been cut out. Okay. But you can see these nucleotides are actually required to get the splicing to occur properly. Okay. And then there's also molecules called ribozymes that are catalytic RNA molecules that can also splice RNA. All right. And this actually also was an interesting discovery because it showed that things other than proteins could actually carry out catalysis. And in fact, there's a prevalent model now that life actually originated as RNA, and that all of the enzymatic activities we're used to thinking about that are carried out by proteins were actually originally carried out by RNA molecules. And nowadays, we don't see them doing nearly as many things as we see proteins doing. Okay. But originally, that was all ribosome-based catalysis. Okay. And there's a couple things about the RNA that let it function as an enzyme. And it's a lot like proteins, right? They can form three-dimensional structures, and that some of the bases in RNA contain functional groups that can participate in catalysis, and they can hydrogen bond with other nucleic acids. Okay. And so I'm sure if you take a more advanced class, you'll learn a lot more about RNA catalysis, but that's all we're going to talk about here. Okay. So what's the importance of introns, though? Why do you waste all this space, then, and how these sequences you have to cut out? So some of those introns contain important regulatory information about where genes should be on and off. Again, we'll talk more about that on Monday. And it turns out that there's another process that can go on. If there are multiple introns, then you actually have alternative ways of splicing the RNA together, and you can actually generate multiple proteins, then, or multiple forms of the same protein. And this is called alternative RNA splicing, and I'll give you some examples in a second. So that means then that a single gene can actually generate multiple different proteins, by changing the pattern of the splicing. So for example, in us, we can have a gene that's active in 2 or 3 different cell types, but it'll actually make a slightly different protein in those cells, not because the DNA sequence is different, but because the cells splice the introns out a little bit differently, which ones they leave in and which ones they leave out. Okay. And it also turns out that in some occasions, those exons actually are sort of protein domains. And what that allows evolution to do is shuffle those exons as units, okay, and put together new proteins. Okay, but that's over long evolutionary time. Okay. So the process looks something like this then. So if you have, let's say in this hypothetical setup, we have 2 introns, we have these 3 exons, and in some cases what really happens is that each exon is a different functional domain of the protein. Okay? And what this allows evolution to do by recombining parts of the DNA is to create new combinations of these functional domains. And by keeping them as separate exons, it makes it easier to mix and match the pieces. Okay? And then the other thing it allows you to do is to make a lot of different protein molecules out of that same gene. So here's again a situation, we've got 3 introns here, 4 exons. And so you see that this may be one way we splice it, okay, and we get this molecule out. Right? But if we do something like we now skip this exon, right, and so our splicing now goes from this to this, then we have a slightly different protein molecule. Okay? That may be missing a particular domain now. Right? Or we can even do things like have alternatives. So we can make, one time we can use this junction, we can use this exon, this one, and skip this one, and then another cell we might skip this one. So we can make 2 different forms of the protein, right, that are the same at their ends, but in the middle have different functional sequences. Right? So a lot of different things can go on. You can even have other ecliptic splice acceptors you might not have noticed, and you can splice into what you thought was an exon and continue from there. So there's a lot of possibilities. And cells actually do use this quite a bit. And some genes have remarkable numbers of intron exons. And this one's even not very high, but it shows you what you can do. This is alpha-tropomyosin. So this mediates the interaction of actin and myosin. And so there's a lot of muscle forms, for example, but they end up having slightly different functions in different types of muscle. And so you can see that how they're generated is by various alternatives in the splicing. So again, in this message, you have what we call a 5' UTR that's before the start codon, a 3' UTR that's after the stop codon. So those aren't actually going to be translated. Okay? And then you've got all of these different possible exons and introns. And even you have alternatives for the 3' UTR that exist as different splice forms. Okay? And so we can end up making a couple different forms. You even have a form in the brain. It doesn't obviously have muscle in the brain cells, but it mediates the interaction of actin that's used for the growth cones for cells crawling. And so this is just an example of how tropomyosin is organized in muscle. But you get various forms of tropomyosin in different types of muscle cells, and that in fact mediates the muscle being different from each other, okay, in their properties, in their physical properties. Okay. So that's how you make the RNA, but now we still have to go and make protein through the process of translation. All right. So in order to do this, there's another RNA molecule we have to introduce you to, and that's called the transfer RNA, okay, tRNAs. And tRNAs are what actually transfer amino acids to a growing polypeptide chain. So now this processed mRNA is going to go and be coupled to a ribosome, okay? And the ribosome is the sort of very large machine that's going to read that RNA and turn it into protein. But there's another RNA, this tRNA, that's involved, and tRNA is what's bringing the amino acids to the ribosome, okay? And what the tRNAs do is allow those codons essentially to be read. Okay, so this is what the structure of the ribosome kind of looks like as a schematic. Okay, and the RNA molecule is passing through this ribosome, and the ribosome is pulling it through. And in a sense, it does it 3 nucleotides at a time. Okay, and these tRNAs bring in the specific amino acid that matches that triplet. Okay, and we'll describe how that happens in 1 second. And what's going to happen is that as it moves along 3 nucleotides at a time, that newly arrived amino acid is going to get added to the growing polypeptide chain, and everybody's going to shift over one, and then the old tRNA is going to leave. So how do the tRNAs bring in different amino acids? So it turns out that the tRNAs aren't identical to each other. Okay, and each one carries a specific amino acid on one end. Okay, and on the other end, it has what's called an anticodon. It has 3 base pairs. They're complementary to a triplet codon. Okay, so you see what's going to happen? So that's going to have a triplet that's complementary to so for example, it's complementary to an ATG. Okay, that means at the end of that tRNA is going to be methionine, because that's how it's going to have the specificity to bring that amino acid in. Okay, so the tRNA is a single RNA molecule about 80 nucleotides long. Okay, and if we sort of have a flattened view of it, it looks like this clover leaf. And so here it has what's called the anticodon. So that's the triplet that's complementary to triplets in the mRNA. Okay, and at the other end, it has a place to attach an amino acid. Okay. And so what happens then is the first thing you have to do is attach the right amino acid based on this anticodon. And so there's actually an enzyme called aminoacyl tRNA synthetase, okay? And it's able basically to match the RNA on the anticodon sequence, and then attach the right amino acid to it, okay? And because there's this flexibility at the third base pair, okay, it's called the wobble position. And so some tRNAs, right, then can bind to more than one codon. This is part of the reason that for some positions, the third nucleotide doesn't matter at all. It can be any of the four. It'll still be the same amino acid. That's because there's not really the tRNAs for different ones. They just come in, and it's just the first two nucleotides of the anticodon that have to match, okay? So you've got this aminoacyl tRNA synthetase, and that's what's actually basically reading that codon and then bringing in the right amino acid. And so it generates what's called a charged tRNA. So charged tRNA is one with a particular, has an anticodon, and it's got the right amino acid attached to it. So your cells are full of these charged tRNAs, right, that have the amino acid with the appropriate anticodon. And what's going to happen then is the ribosomes are going to facilitate the coupling of the tRNA anticodons with the messenger RNA, okay? And that's going to then generate a specific polypeptide chain, okay? So the ribosome has two subunits called a small and a large subunit, and they're both made up of both protein and RNA, okay? One interesting thing is that a lot of antibiotics you use take advantage of the fact that bacterial ribosomes actually have some important differences from eukaryotic ribosomes. So a number of antibiotics, what they actually do is target the ribosome, but they don't bother your ribosomes, but they interfere with bacterial ribosomes. So they shut translation down in bacteria, but they are not of any harm to you, and that's how they end up working. Okay. So this again is an image then of what's going on with the ribosome, just slightly a more realistic protein structure view in a schematic. And so it's got basically three places to bring in these, or three positions that tRNAs can be. Let's see if I can slice it in here. Yeah, I'm just going to skip to this one. Okay, they're called EP and A, and what's going to happen is they're going to be sequentially filled as the whole process is occurring. So you're reading these mRNAs coming through here, and then it's read as triplets, because what happens at any one moment is that you have a tRNA that comes and it matches this particular triplet, so it's got the right amino acid. There's already a growing polypeptide chain. So this was the last tRNA, the previous tRNA that had come in, okay? And this one is one that's now exited. So what's going to happen is that this amino acid is going to be attached here, okay? So the amino acid chain is going to grow. Then everything is going to shift over one. This one then is going to, this chain is going to be transferred here, linking it to this amino acid, and then this one's going to shift out and go out, and then the next triplet is going to come in, and the next tRNA is going to come in, it matches it, brings in the right amino acid. And so that's how the ribosome is going to work. So it's going to chug along, basically able to look at every three base pairs. The right tRNA comes in, and the right amino acid gets added to the growing polypeptide chain. Okay. And so this is what I just described to you. That it has the three binding sites for tRNA. The P site holds the one that's going to come in, that's growing, sorry. The A site holds the next one that's going to be added, okay? And then the E site is where it's discharged. All right. Okay. And during the elongation stage, right, amino acids are added one by one, and they're added at the C terminus. Yeah. That's right. It's based on the mRNA sequence that you would see, and yeah, you think about it, that's what it's based on, not the anticodon sequence. Yeah. By convention, we write it out so that, the start methionine is written as the ATG, and then you can follow it. It gets confusing when you sequence DNA and you're trying to figure out which one you're reading. Okay. But to simplify it, it's written that way. It's actually then the template strand. I mean, it's as though you're reading the template strand of the DNA too. Okay. So translation proceeds along the mRNA 5' to 3', so it has a directionality. And then that also means that your protein grows from N to C, from the N terminus to the C terminus. Okay. Okay, and so what ends up happening then is that this cycle just keeps going over and over again. So you initiate, we'll show you that process in a minute, at the start of methionine, and then you just keep growing it and growing it. Okay. So again, though, that's the elongation phase, but you have to initiate it and you have to terminate it. Okay. And so there are various ways that you initiate and terminate. So initiation brings in the first amino acid, and in pretty much every case, right, that's a methionine. So that's the start codon. Okay. And it binds with a special initiator tRNA that recognizes the AUG and brings in methionine. Okay. And that completes the, in fact, assembly of the whole complex. It's going to continue elongation. Okay. So initiation begins by bringing in, right, so that's the AUG, the anticodon is UAC, okay, and that's going to come in and be the methionine, and then that actually is what then assembles the rest of the ribosome onto that. And then the process can get going, and then you can bring in the next tRNA and so on and keep on going, and that's how you initiate the translation. No, it's not, right? That's why there's redundancy in the code. So that's why you can have ones that, for example, for certain ones, the third nucleotide position won't matter. Okay, so termination comes when it reaches one of the triplets that's a stop codon, okay? And when it does that, then it brings in this thing called an A factor, or it reaches the A site, and it brings in something called a release factor. And then that causes an addition of a water molecule at the end of the growing polypeptide chain instead of another amino acid, and then that stops it, right? And so then nothing more gets added, and the chain terminates. And so there's those three stop codons, right? And so if any of those are encountered, then this thing called release factor comes in, it adds the water, and then the ribosome comes apart, and the chain reaction, the process stops, okay? It turns out that you can, in fact, again, start translating an RNA with multiple ribosomes. So ribosome can get started and go a little ways down, and another ribosome will assemble at the start AUG, and then get started, and so you can have a chain of ribosomes going along. So you can actually then be generating simultaneously a bunch of different polypeptide, or a bunch of copies of the same polypeptide chain off of the same mRNA molecule. So this lets you ramp up pretty quickly. And so you can get these strings of ribosomes going down. Okay. And then it turns out that translation sometimes isn't all you need to make a functional protein, and there's other modifications that occur. And there's really, again, it would be a huge effort to tell you all the different modifications that can occur, but the book goes into a few of the what we call post-translational modifications. So the protein has to fold, and there are certain modifications that then can affect the folding or modify the activity. And so these are just a few. So for example, some polypeptides are made, but then they have to be cleaved by something else in order to be active. Okay. A good example is insulin. Insulin is made as a long chain, and a middle section of it actually has to be cut out to make the active molecule of insulin. Okay. So that occurs post-translationally. Right. And then sometimes the polypeptides actually are separate genes. They have to come together to make an active protein. Okay. And then another thing that happens is that proteins can be targeted to certain places. And one of the things that you see right away is that some proteins are bound to be in the cytoplasm, and other proteins are supposed to be on the membrane of the cell or secreted from the cell. And so that, in fact, is initiated by sending the ribosome to different places. So free ribosomes that are in the cytoplasm, the proteins they synthesize then are free in the cytoplasm and stay inside the cell generally. But if the ribosome gets bound to the membrane of the ER, okay, then it can actually be targeted to either be a membrane protein or get secreted out of the cell. All right? So that's one of the things that you see. And there's actually a signal sequence that starts off proteins that are supposed to go to the ER, the endoplasmic reticulum, for secretion. Okay? So if they don't have that signal, they stay in the cytoplasm. If they have that signal, what happens is that part gets translated, the ribosome actually will pause, and that thing will dock to the ER, and then the growing polypeptide chain will end up in the ER either as a transmembrane protein or being secreted. Okay? And this just shows that bit that gets made first, the signal peptide, gets bound by another protein called the SRP that's in the cytoplasm, and that brings it to the ER. So you have this process for some ribosomes where they start to make the signal peptide, then they go to the ER, and then they secrete the rest of the polypeptide chain into the ER so it can be secreted. Okay? I guess we'll end there. Okay?